you suspect that, it's a whole totally different realm of antibiotics and medications that you've got to administer to someone if that's the type of microbe they have. You also have a group called the Mycoplasmas. So these are called the Mycobacteriums, ones that have the mycolic acid layers. You also have the Mycoplasmas, and they're different as well. They actually don't have a cell wall like we've typically just learned about. They have a cell wall that's made up of lipids and sterols, and so it's just a different composition. We're still labeling them as a bacteria, and but they are different, and it just, one of the differences that we've come across. You've got a term down here that's thrown around, pleomorphic. We'll see this again when we look at our yeast. Yeast, they label them a little bit more like dimorphic. But pleomorphic, what that means is that these types of bacteria, because of their differences in their cell wall, the mycoplasmas, they can change once they're in your body or in a different environment. They'll adjust. They'll adjust their shape to where they look different. So if you're looking for a round cell that measures 1 micrometer in size, some of the mycoplasmas will look that way, and then others will look more like a rod, and they'll be 2 micrometers in size or 3. And so they change their shape. They're very difficult, frustrating for us because of that. But if the temperature changes, so if they get out in the environment versus in our body, they can adjust and produce different enzymes and things like that. If the pH changes, they can adjust to that as well. They can change their shape, so they're a little different. Of course, there's always exceptions to all of our rules that we come up with, isn't there? But these are your exceptions, the mycolic acid layer and the mycoplasmas. I'm going to pull up our bacterial list for just a moment. And I think one of you had asked me, and I think luckily I can put this on here because we do reference it some. Do y'all remember looking at this a few weeks ago? Mm-hmm. So the mycolic acid, the mycoplasmas, the mycobacteriums. Did I say they were gram-positive or gram-negative? Gram-positive, right? So this is our gram-negative list. Let's go on down here to our gram-positive. And y'all see how they're pretty easy to identify, Mycobacterium. So that means if they have the genus Mycobacterium, that means that they're a bacteria that forms that mycolic acid layer. That they will have that mycolic acid layer. And then you have the Mycoplasma pneumoniae here. It causes a very mild form of pneumonia. But look at what all the mycobacteriums do. Tuberculosis, which we also have paratuberculosis, which is mainly in cattle. It's a little bit seen with people somewhat. TB in cattle, so Mycobacterium bovis. Leprae, so Mycobacterium leprae causes leprosy, so this is a mycolic acid layer on this bacteria. Avium is one that we see like as a secondary infection in AIDS patients. And then Kansasai, right? This is mainly relegated to Caucasian males who have a pre-existing condition, usually emphysema, bronchitis, so some type of lung issue. And then you've got some others here, but do y'all see how you can identify them from the genus? So if we test, we have a patient sample, we test, and it turns, and it tests gram-positive. Depending on what kind of symptoms they have and what you're suspecting, it may be necessary to do an additional mycolic acid test to see if you have a bacteria that forms a mycolic acid layer. If you do the initial test and your bacteria is gram-negative, would you suspect a mycolic acid layer then? No, because this only occurs in gram-positive bacteria. Okay, so if y'all remember, we were working outward from outside to inward. So we're all the way to the cell membrane. So we're not quite on the inside yet. Cell membrane looks very similar to ours, right? It's a phospholipid bilayer. So the hydrophilic head, hydrophobic tail, and the double layer. You've got stuff embedded in the membrane. one of the main differences here with the cell membrane is going to be the function of the cell membrane as far as the additional functions that it has to do. Of course, it's controlling what's going in and out of the cell, just like ours is. It's doing signaling and transport and all the same stuff that our cells do. But do y'all remember that the prokaryotic cell has no organelles, no membrane-bound organelles? It's got ribosomes, which are not membrane-bound, but it's got ribosomes. It's got DNA. It's missing all of that other stuff that has jobs. The Golgi body, the rough and smooth ER, the lysosomes doesn't have any of that. So all those jobs that those organelles do in our cells, the bacterial cell still has to do all those same jobs. Still has to digest and break down, lipid synthesis, protein transport, all of that stuff. But its membrane has to do it. The prokaryotic cell membrane has to do those jobs because there's no organelles to do them. And they give us a picture. Looks very similar to what you'd see if you looked at a picture of ours. Different things embedded in there. So, if there's no organelles inside the cell, no membrane-bound organelles inside the cell, what is inside the cytoplasm of the bacterial cell? It's mainly water, right? 70 to 80% water, and then the rest of it is just some nutrients. You've got some sugars that are brought in, some amino acids, which are proteins that are broken down, some different salts, so almost like electrolyte stuff going on there. But mainly it's water, which is just very similar to our cells. You also have some genetic material inside the cell. Now we're not discussing viruses yet, we're on bacteria, and so they still follow the rules. DNA is still DNA, RNA is still RNA, it's all very similar. Really the only main difference is going to be the shape. With us, we have 23 pairs of chromosomes, so 46 chromosomes, and they're cut, and we actually have one big long strand and it's cut into 46 pieces, right? With the bacterial DNA, they have just one circular piece. Just one circular piece of DNA. So we call it just one chromosome, is what we'll call it. They also have an extra set of DNA. So see if there's a picture here. Not a good one, that's. We're looking at a bacterial cell here. You'll have the DNA. I have just the single circular double-stranded typical DNA molecule there. But then elsewhere in the cell, you're going to have what's called a plasmid. And this is where you see a little bit of variation among different microbes, among different bacterial cells, even in the same species. The plasmid is the extra stuff. So the DNA, the one chromosome, the one circular chromosome, has all the necessary stuff. It had the bacteria has to have everything in that DNA chromosome to function. That's its code for being. But the plasmid has extra stuff that if a bacterial cell comes across another dead cell somewhere and it can pick up genetic code and incorporate it into its own plasmid, or if we give it the green fluorescent protein out of a jellyfish or something like that, it will incorporate any of that extra stuff that it picks up into this plasmid here, and it just holds on to it, and it will share this genetic information that's in this plasmid with other bacterial cells. Remember we talked about conjugation, those tubes that form. Viruses, when they, there's some viruses called bacteriophages, they only infect bacterial cells. And they will inject their DNA, RNA, whatever it is, into the plasmid of the bacterial cell. And the bacterial cell will still read it and do what it says, but this is where it keeps all this extra stuff that it picks up. So it's really neat. So when we modify bacteria, or when we genetically manipulate them in any way, we're manipulating the plasmid, not the essential DNA here. Unless we want to harm them, right? And if we're doing it for a pharmaceutical benefit, plasmid. that really wasn't said on that slide that I do want to point out is that, and we've said it already, but there are no membrane-bound organelles within the bacterial cell. So what that means is that the DNA is not surrounded by a nucleus, right? There's no nucleus here. There's still a spot where the DNA lives in the cell. It's called a nucleoid, which is the title of that slide. And there's a spot designated for the DNA to sit, but it's not surrounded by a membrane. So it's not actually called a nucleus since there's no membrane surrounding the DNA. The ribosomes. So we're inside the cell. We've gone from the appendages for movement, one's for attachment. We've gone through the layers of the cell wall. We talked about the cell membrane. We looked at the DNA. The only other thing in the cytoplasm, excuse me, the only other thing in the cytoplasm of real significance as far as this class goes is going to be the ribosomes. Ribosomes, they're an organelle that we have too. Do y'all remember what the job of the ribosome is? Protein synthesis, right? Same job with your bacterial cells. The ribosomes are a very common one that we will target with a drug because it's something that they have. It's also something that we have, so we have to be careful about that. But there was a researcher who found out that the ribosomes that bacterial cells have seem to be a little different than ours. We both have ribosomes, do the same things, protein synthesis, but there is a man, I can't remember his first name, his last name is Svedberg. And he won a Nobel Prize in chemistry for his work with this. Svedberg, and I'm going to write that up here just because. Svedberg. And he noticed that when he would take the contents of a bacterial cell, which included the ribosomes, and then take the contents of our cells, our ribosomes, and he would spin them in a centrifuge. Do y'all know what a centrifuge is? So it's something that spins really fast. It can separate out different particles, separate out blood from the plasma and all this stuff. So he spun them in a centrifuge, and what he noticed was that it seems very straightforward, I guess, and I'm gonna goof it up, but basically there's a unit of measurement called a Svedberg unit that he came up with, and it's a unit of measurement of time. And the time is measured in how long it takes for a piece of sediment, something like a ribosome, to separate and go to the bottom of the tube in a centrifuge. So how long it takes it to fall to the bottom of the centrifuge. It's called a Svedberg unit. Well, he noticed that there was different amounts of time that was occurring for the bacterial ribosomes versus ours. When you spin them down, they would fall at different rates. And so he analyzed that. Now, what does that mean for us? Well, a ribosome kind of looks like this. If we draw it, typically it's just one big circle when we draw a ribosome. It's the little dot that we always see in the cells, but it's actually made up of two separate units. There is a top large subunit and then a bottom small subunit. So I'm going to do large, small. Start with ours first. Our ribosomes are an 80S ribosome, so eukaryotic. We have 80S ribosomes. It doesn't mean anything yet, right? The S is Svedberg units, so it's the amount of time it takes for it to fall when it's spun down, 80. For a prokaryotic cell, they have 70S. 70S. Now, the reason I'm even explaining this is because when you look up here, it says that it's 60% and 40%, and then we get a 70S. Doesn't make any sense. What he means by this is that when he spun these down, and these will spin as separate units, the prokaryotic ribosome, it fell at a rate of 60S. The smaller part fell at a rate of 40S. Together, when they stayed together, they fell at a rate of 70S. 60 and 40 don't add up to 70, and that confuses people. But what this actually means is that if you were to spin them separately, this is how long it takes this unit to fall, this is how long it takes the bottom unit to fall. If they stay together, this is how long it takes them to fall. Ours were a little bit different. So our, as one big unit, the eukaryotic one, falls at 80 S. Let's see here. I think I have a picture. Oh, I thought I gave y'all the wrong numbers there. 50 and 30. Ours are 60 and 40. I'm sorry, I told you I'd pick that up. 50 and 30. Okay. Again, it does not add up to 70. The prokaryotic ribosomes fall at these rates, 50 and 30, and then together fall at 70. Ours fall at 60 and 40 to give us an 80S total. So, because he noticed these differences, it must mean that they're not following at the same rates. It must mean that there's something different about them. And there is. Molecularly, they're a little bit different, and we know that now. And so we have a drug that we can give someone that targets the protein synthesis. It targets the ribosomes of the bacterial cells. And when we give it to a person, it doesn't bother our ribosomes. It'll mess up the ribosomes for the bacterial cell because they're a little bit different. It targets that difference. But it doesn't seem to have any effect on our ribosomes, which is great. Because if you can keep a bacterial cell from synthesizing the protein that it needs, it'll eventually have a problem. Either it won't be able to reproduce, or it'll be metabolically inactive, which are things that we associate with cell death, something. So you're affecting it in some way. But because of those differences, we're able to figure that out. So when you see those numbers and they don't add up, that's why. It's talking about the relationship between the two units. Okay, and then inclusions and granules we don't really do anything with in here. Bacterial cells have them. They're like little storage tanks. But they're nothing that we test for or check for or anything like that. We don't do anything with the cytoskeleton, y'all skip that. Endospores. So this is listed as an internal structure, even though what the endospore is it's a hard layer, a hard, very durable, very protective layer that will form around the genetic material of the cell, and then all the rest of the cell will shed off, and you're just left with this hard endospore. It's considered an internal structure because it forms on the inside, even though it ends up being an outer layer. So let me show you all a picture of this, and then we'll come back to this slide. This is what it looks like when the endospore is forming. So let me stop here on a word, vegetative. Vegetative has a very negative meaning when you talk about people. When you're talking about a person in a vegetative state, that is very different than talking about a cell in a vegetative state. A cell in a vegetative state is its normal, functioning, metabolically active, reproducing state. So it's the way it's supposed to be, right? The vegetative state for a cell is a good thing. And then, if you have a cell that goes dormant or forms an endospore or something like that, it can sporulate or germinate back into a vegetative state, which again is a good thing, right, as far as the cell's concerned. Now, if you follow the numbers through here, you can kind of see what's happening. So this is just a rod-shaped cell with its one circular strand of DNA here. The DNA will copy itself, right? So the cell will make a copy of the DNA. A layer, a protective layer, will form around the copy of the DNA that was made, and it keeps forming, and it ends up being a spore that forms a very calcium rich, very dehydrated, so not very much water as part of the spore, just protective layer that forms around the DNA. Once that layer has finished forming, the rest of the cell sheds off, and now you have an endospore. When do these form, and what's the purpose? Well, we only see them form in a gram-positive cell type, so we have not discovered them in any of the gram-negatives yet. It seems to only be gram-positives. Clostridiums, the bacilli, which bacilli is the one I've got for y'all, your endospore-forming microbe to work with this week. And then some of the sporosaurs will do it as well. But the endospore is something that the microbe will do, the bacteria will do when its environment changes. So why it's a concern for us is because when you consume an endospore-forming bacteria, it won't have formed an endospore when you consume it typically. Sometimes they have. But if you consume one in a vegetative state, it will form an endospore as it passes through your really acidic part of your body. And then once it's out of the acidic part, then it will go back to its vegetative state and start to set up camp. And so it forms a protective layer as it passes through your harsh parts. We've also noticed that they'll form an endospore out in the environment. So remember, not all microbes are bad for us. Some are just out in the environment doing their thing, but we have discovered some that have done it because their pH changed, their environment changed. Some of them, their trigger is that the nutrient that they need is not present in the environment anymore. And then when you give them that nutrient back, they'll come back to a vegetative state. So we see them doing it for very different reasons. The endospore is very indestructible. So there's no antibiotics, not necessarily any chemical that you could safely give a person that's going to target the endospore, and so they're very hardy. We have been able to, like on equipment, surgical equipment and stuff, utensils and things. My autoclave's not here anymore, but autoclave, so high pressure, high temperature, steam, does seem to have an effect on the endospore if you do it for a long enough period of time, but you can't do that to a person, right? You can't do that to a person. But our drugs, our dermicides, disinfectants don't seem to really be that effective. How long can a microbe exist as an endospore? Seems to be indefinitely. There is a bee, as I said, a bee, B-E-E-B, that we isolated an endospore from. Is a fossilized bee. It was 25 million years old. They pulled the endospore out of it, hydrated it, gave it nutrients, and it came back to life, right? It came back to its vegetative state, so it was just dormant. 25 million years is what they were dating it. They also found a type of bacillus, which is one of the ones we'll work with again, but a type of bacillus up in the ice in the Arctic region. 250 million years was the date on it. They were able to get it to come out of its endospore back to a vegetative state. So how long can they do this for? Do not know, right? It seems to be indefinitely, as far as we can tell, but probably we're going to use Bacillus cereus in here, which is like if you were to swab this tabletop, there's Bacillus cereus on here. So it's a real common one with food poisoning, like from home. Set something on the counter and then you don't cook it real good afterwards. It's just one that'll show up there. As long as it stays where it should be, it's not a problem. But the other one is Bacillus anthracis, so anthrax, and that was, a bioterrorism scare for a while there. They have an island. Let's see if I can pull it up. It's called Anthrax Island. Have y'all heard of that before? Anthrax Island. There is an island where during World War II, well actually before World War II, that they tested these anthrax bombs on this island off the coast of Scotland, I believe, and here's some pictures. And so they dropped these bombs on this island, and of course there was nothing alive there after they did that. Well, it's an endospore forming microbe. How do you clean that up after you've dumped an anthrax bomb on an island? How do you clean that up? It's going to form an endospore. Well, they used some very heavy concentrations of formaldehyde and several other compounds, and they say they've got that island cleaned up. Nobody's willing to go live there yet, but they keep a couple of different herds or flocks of sheep that stay and live on that island, and they're monitoring them, seeing if they ever contract anthrax. But it's Anthrax Island, and it's because of the endospore. They're just, how do you get rid of that? And who's going to believe you that you say that you have? Heat, drying, freezing, radiation, chemical emission, most of those things, it just seems to be very resistant. So it's definitely, if you have a microbe that can form an endospore, you want to know it. So it is one of the staining techniques that y'all are doing next week, is being able to identify if you have a microbe that has formed an endospore. And there's probably the hang up. It doesn't tell you if you have one that can form one, just if you have one that has formed one, that has already formed an endospore. One other thing to kind of tuck away in your brain as far as just thinking about this is, for a microbe, for a bacteria here that forms an endospore, it takes about anywhere from 6 to 12 hours to form the endospore. But once the endospore has formed and the bacteria is ready to go back to a vegetative state, it can go back to a vegetative state in about 2 hours. So it's a lot quicker to go back. Okay, the last real thing I want to touch on here is some shapes. And we've already talked about this a little, but it's kind of helpful to get a visual. It used to be one big table. Where's my big table go? We'll just do individual ones. So we've looked at some of these shapes of our microbes. So bacterial cells, they have many different shapes. Round, rod-shaped, spirals, and round is, as you can see here, round is round is round, right? There's several variations to round. So coccus is our term for round. So if you see that in a bacterial's name, that's referring to a round-shaped cell. But again, look at all the different variations of round we have here. This one, I don't know that I would automatically identify it as round. Sometimes you might think that more rod-shaped. That's weird looking as well. But these are all considered round coccus shapes. Rod-shaped has the same problem. Look at all the different variations we have for what we're calling rod-shaped. I think the general consensus with rod-shaped is that it's longer than it is wide. Then you've got something that would be rod-shaped. But you've got skinny ones, plumper ones. So when you're looking under your microscope, especially at these live cultures, not the demo slides, but the live cultures, you may see several of these on one slide. It doesn't mean you're looking at a bunch of different things, just cells kind of take different shapes sometimes. You're going to see some variation in that. Vibrio is got some curves in it. Now, look at the next one compared to Vibrio. Spirillum, also curvy, right? Spirillum's also curvy, and spirochete is also curvy. So you've got several here. The Vibrio looks more like a rod shape, and it will typically only have one slight curve to it. So it's going to look like a rod-shaped bacteria, but not straight. It's going to have just a light curve to it. Cholera is one that I have a demo slide of later on that we're going to look at that has this shape to it. Some of them will have flagella, some won't, so that can't be an identifying factor with them. The spirillum are rigid. That's one of the ways we differentiate them from the spirochetes. The spirochetes here see how they look very similar to the spirillum. You've got quite a bit of wavy spiralness going on there. But the spirillum are very rigid, and they're not going to be bent in any way as far as their overall general direction. Whereas the spirochetes, see how you got some, you can tell that they're a little more flexible in how their shape is. The spirochetes will not have a flagellum on them at all. None of the spirochetes will. wanting y'all to get off of this is that when you see something under the microscope, you're gonna see some variation, and that's okay, right? Rod is gonna mean several different things, round's gonna mean several different things. Even the spirochetes and the scrollum, they're gonna look several different ways, and it's frustrating when you're trying to learn what you're looking at. It's very frustrating, but that's okay, right? It doesn't mean you're not seeing what you should be seeing. Okay. Now, got some other terms that get thrown around as far as shape and arrangement go with our microbes. So some of these we, again, already talked about. What did cockeye mean? Round. Round. Cockeye's round. So when you see round cells under the microscope, when you're looking at bacterial cells, sometimes you're going to see single individual cells just by themselves. Sometimes they're going to pair up, and so we have terms for all that stuff. Diplo or di means two, and then cockeye's round. So this is a pair of round cells. Tetrads are groups of 4, so you typically don't see them in odd numbers, because when a microbe replicates, go from 1 to 2, and then 2 to 4, and then 4 to 8. Y'all see how it would always be an even number once you don't have just one? Because you're always copying everything that's there once they replicate. And so it's typically always going to be we're labeling them off of even numbers. So cockeye's round, and then you've got some terms you can apply to that. And then irregular clusters, your term for that is staple, means irregular clusters. Staple means irregular clusters. So if I have staphylococcus, what would that mean?
Irregular rounds.
Yep, so you're looking for round-shaped cells that are in irregular clusters. Round-shaped cells that are in irregular clusters. What if I had Staphylococcus? What would that mean? Do you remember what bacilli was? Rod-shaped, right? Bacilli is rod-shaped. So you have irregular clusters of rod-shaped bacteria. Chains is strepto. Strepto is chain. So if you had a streptobacilli, what would that mean?
A chain of rod.
Yep. A chain of rod-shaped bacteria cells. Or a streptococcus would be a chain of round-shaped cells. The palisades, I want to show you all a picture of those because they look like columns in a palace, and that's, so with your rod-shaped bacteria, we typically think of them linking up like their ends, but sometimes they'll stack sideways, and it looks like columns. It's called a palisade arrangement. So let me, find a decent picture here. That is an okay picture. Let me get an actual, here. Y'all can't see that very well. Maybe a little bit. What happened to just being able to pull up a Google image?
I know. That's so aggravating.
This would probably be without having an actual one to get to pull up. Y'all see how they stack straight up and down instead of end to end? See if I can get it. So right there, you've got several that stack straight up beside each other versus some that are end to end. So the palisade is where they're stacking up and down, side by side like that. Same here, versus a chain of them end to end. Okay. And our last slide item that we're gonna cover out of chapter 4 is just the size of bacteria, which I've already measured yours, but we haven't really measured them in relation to anything that we've probably encountered before. So let's take a look at this. So, they did a decent job with this, right? Human hair at 200 magnification, and then you've got your pollen, bacterial cells, blood cells, all that stuff. So, red blood cells are probably something y'all looked at before, I think so. Red blood cells are about 10 micrometers in size, so ragweed pollen, which is going around right now, right, is a little bit bigger. Your lymphocytes, so immune function here. Yeast, we looked at yeast, right? They were some of our bigger ones, 2 to 10 micrometers. And so if you get down here and look at your red blood cells and pull the yeast out of the picture, you've got Ebola virus. E. coli here, see how big it is compared to virus. Staphylococci bacteria, so they're a little bit smaller than E. coli. The rhinovirus, which is responsible for colds, right, common cold. And then you see how much smaller the viruses are. If you put this into terms that we can think more of, if you took one of our cells, it would be about the size of a football. If you took a bacterial cell in relation to that, it would be, let's see, it'd probably be about the size of like a Tylenol or some kind of medication, a pill. And then a virus would be if you took a marker and put a dot on one of those pills. So there's your scale, football pill, and then a little marker of a dot on there. So it's quite a bit smaller. Okay. You have not probably ever talked about a bacterial cell before we went through this chapter. Do you have a little bit better of a visual in relation to what a bacterial cell is and what bacteria are in general? Because they're just a cell, right? That's what they are. And they do all these different functions. Once we get further on in the semester, we'll look more specifically at what makes them such a problem for us. Because they are that basic cell type, but then they all have like an enzyme that they can produce that makes us very sick, or they have this particular toxin that they produce, or this particular protein that our body is allergic to, or something like that. So they all have their thing that they do, and that's where we'll start picking out some of the differences. But for now, that's your general what a bacterial cell looks like. And so there's some similarities to us in there. Okay, do y'all have any questions? Okay, let's move into our lab stuff now. If y'all need a break, you can step out and take it. One of the things I want y'all to do first before I explain the lab is we're going to be using microscope slides. So I've got boxes of them up here. I'd like for y'all to go ahead and take your slides and put them in these Copeland jars. These Copeland jars have alcohol in them. We're trying to maintain aseptic technique, right? We want our microbe that we put on our slide to be the only thing that's on our slide. So we're going to disinfect our microscope slides. The alcohol helps to wash off. It breaks down the hold that a cell would have on a microscope slide, and it'll just come right off into the alcohol. So I'd go ahead and get your slides soaking. They can be soaking while we're talking about what we're doing, and then they'll be all ready for y'all when you get to that point in the lab. Let's see. I would, how many are in here? 1, 2, 3, 4, 5, 6, 7. Yeah, I would get 2 of these per person, and then like if a couple of you are working together, just do, one pair of you's gonna have to have just 2 together. But everybody else get 2 to yourself and fill it up, because you're gonna use a ton of slides. You're gonna have to refill these once we get started. But I'd fill all of these up. Y'all can use them in one big class or however you want. They're probably all need to be filled up. But while we're taking a break and shifting, y'all can do that.